Yeah, so my name is Joran Hendricks, and I'm a recent graduate from Wageningen University, and I'm currently affiliated with the Borneo Nature Foundation International. For my research, I focused on bioacoustics of gibbon songs in central Borneo, and I was looking at a population in central Borneo where you have two species, the white-bearded gibbon and the Muller's gibbon, which they overlap in distribution ranges, but there's also believed to be a natural hybrid gibbon. Um, I use bioacoustics for the main idea is that it's a non-invasive technique of actually studying animals that are very hard to see, that are cryptic and extremely arboreal. I use three different techniques: passive acoustics, active acoustics, and triangulation, because each method has their limitations, but together they actually compensate for each other's limitations. A main advantage of using bioacoustics is, especially for extremely arboreal or cryptic species, is because you can hardly see them. Morphological determination is challenging, and as a result, also collecting genetic samples is quite hard. But with bioacoustics, since these primates, they sing very loud songs early in the morning, and you can hear them very easily. Although you don't see them, you're still able to survey them, you're able to monitor them, and you're able to study them. My main focus of this research was to find a more cost-effective and efficient way to study gibbon populations without having to see them or without having to genetically sample them. A main benefit of this is that just through acoustic analysis, you can actually infer vital conservation metrics, although that was not my focus of my research, but it lays the foundations to do that in the future. What I did for my research is that I focused on the specific vocal fragment called the Great Call, which is only sung by the female gibbon early in the morning. If you look at it on acoustic software, it's very easily recognizable. It has a couple of notes, and what I tried to do is that I tried to describe that call into 10 different parameters. And then using those parameters, I tried to identify, since gibbon songs is genetically determined, genetically expressed, and it's genetically conserved, although it does have variability. The idea was to see whether this can be used as a more cost-effective way to basically replace morphological identification and the need for detailed genetic analysis.